Hello students. So today we are going to study about meiosis 2. We already discussed about meiosis 1 and we know the result of meiosis 1, from one cell there is formation of such type of two daughter cell, and in each daughter cell chromosome number it is reduced to half. We know that parental cell consists of total 4 chromosome, and in after meiosis 1 there will be formation of two daughter cell from one parental cell, and number of chromosome it was reduced to 2 in each cell from 4. So in this way, that was the result regarding with meiosis 1. Now student, after meiosis 1 there will be meiosis 2. Meiosis 2 and meiosis 1, it is now interrupted by a short resting phase. We can say it is interkinesis. So interkinesis, it is the phase which will exist in between meiosis 1 and meiosis 2. That interkinesis, it may be now also short, it may be long, sometimes absent also. And interkinesis mainly results into formation of protein synthesis and RNA formation. So in interkinesis there may be formation of RNA, there may be protein formation which is required for further steps, but there will be no DNA duplication. There will be not duplication of genetic material. Okay. So in this way, earlier we already clear that the genetic material, that was the DNA, it become duplicate only once. Okay. So there will be no duplication of DNA or genetic material during interkinesis. During interkinesis, there will be only formation of RNAs. There is the formation of proteins. So after interkinesis now, there will be meiosis 2, meiosis 2. Meiosis 2, it is also divided, we know, into prophase 2, metaphase 2, anaphase 2, telophase 2. Okay. And after telophase 2, that is karyokinesis 2, there will be cytokinesis 2. Okay. So from prophase 1, we have to study that particular phase. Now students, phases of meiosis 2, it is very similar to the mitosis stages. Okay. So we already studied about mitosis, and from one cell there will be formation of two daughter cells. Their number of chromosome it will also equal during mitotic phases. Okay. Meiosis 2 phases of meiosis 2, it will be now very similar, exactly same to that of stages of mitosis. Okay. So here we know the result of meiosis 1, that is the two cells. Okay. And in each cell there is the presence of two daughter cells. So after completion of karyokinesis of meiosis 2, there will be formation of again two daughter cell here. From one there will be two daughter cell, and chromosome number here there is 2. Again after completion the chromosome number it will be remain same. So students, meiosis 2, it is exactly similar to mitosis. Okay, where one cell it will be divided into two cells having same or equal chromosome number. Okay, so that is about the meiosis 2. Now we have to study the stages of meiosis, and we know there is very similarity in all the stages of meiosis 2 with mitosis. So we have to study prophase 2, we know. So in prophase 2, we know chromosome they will become double stranded, dyad condition. Okay. Here the nuclear membrane and nucleolus starts disappearing. Okay. And at the end there will be starting of formation of spindle fiber, similar to metaphase stage of similar to prophase of mitosis. In next stage student, that is in metaphase 2, no, there will be arrangement of chromosome on this equatorial plane. So that is the first daughter cell, second daughter cell. So that is chromosomes, they are arranged on this equatorial plane. There is formation of such type of the spindle, chromosomal fiber, okay, or there will be formation of interzonal fiber, like that. The chromosomes, they are attached here, pole to the centromere and centromere to the pole, okay. So in this way there will be attachment. There will be now such type of arrangement like metaphase of metaphase second. After metaphase, students, we know there will be anaphase. During anaphase, we know what will happen? Splitting. Okay. So there will be splitting of centromere, and due to splitting of centromere, two chromatids, they will separate, and out of that, one it will be move toward one pole, another will be toward another pole. So due to splitting of centromere, there will be occurs, and again there will be shortening of this chromosomal fiber. Okay. Again, second type of the fiber which will be arises in between two centromere, it is interzonal fiber. Okay. So that interzonal fiber, it will now elongates, and there will be shortening of this chromosomal fiber, and in this way there will be movement of chromatid, it will be takes place toward this opposite poles. Okay. So in this way that chromosome, it will be divided into two chromatids and it will move toward opposite poles. So that will typically very similar, anaphase second, it will be similar to anaphase of mitosis. It will differ from anaphase of meiosis 1. Okay. So we know in meiosis 1 there was not splitting of centromere. Okay. The entire chromosome, it will be moved toward opposite pole. So that is anaphase 1. So both the cells, they will be undergo anaphase 1. After anaphase 1, student, there will be the anaphase second, telophase second. So telophase second, we know telophase second में the chromatids, they will be moved toward opposite pole. There will be reorganization of nuclear membrane and nucleolus. Okay. And finally, there will be formation of two daughter nuclei having similar number of chromosome to that of the parental one. Okay. So in this way, there will be telophase 1, there will be disorganization of nuclear membrane, disorganization of nuclear that is nucleolus, spindle fiber formation, it was disorganization. Okay. And in this way, one nucleus, it will be divided into two nuclei at the end of telophase second. Here karyokinesis second completes. Okay. After karyokinesis second, students, there will be again cytokinesis second, and we know during cytokinesis second there will be division of cytoplasm. So here there will be division of this cytoplasm in plant cell by cell plate formation, and in animal cell, cell furrow formation. In second cell also, it will be divided into two cells, and there will be formation of such type of two daughter cell from one cell, here two daughter cell from one cell, and in this way there will be formation of total 4 daughter cells. Okay. So again, cytokinesis, it is very similar to the cytokinesis of mitosis. Okay. So in this way, students, in meiosis 2, which is very similar to that of mitosis, two cells, okay, having here 2n equal to 2, here 2n equal to 2, diploid chromosome number, it is 2. Okay. So that two cells, they will convert into 4 cells, and each having 2n equal to 2, same chromosome number. So meiosis 2, it is exactly similar to the equational division or mitosis. Okay, student. So that was about meiosis 1 and meiosis 2. Finally, students, we have to study about significance of meiosis. First significance of meiosis is for formation of gametes. So due to meiosis, we know in sexual reproduction there is formation of gametes, male gamete and female gamete, which it will be undergo fertilization, form zygote, and in this way maintain diploid condition of the zygote. Okay, so meiosis and fertilization is the steps where there will be maintenance of constant chromosome numbers. Okay, so gamete formation के लिए, okay, क्रोमोजोम नंबर ऑफ स्पीसीज को मेंटेन रखने के लिए, कांस्टेंट रखने के लिए. थर्ड वन एप्लीकेशन वी कैन से स्टूडेंट्स, इफ देयर विल बी एब्सेंस ऑफ meiosis, देयर विल बी फॉर्मेशन ऑफ abnormal tetraploid or hexaploid cells. Okay, so such type of the cells, they will be abnormal. Next one application, student, in meiosis we know one of the very important event which will occur, it is a crossing over. So during crossing over we know there will be exchange of homologous segment of the chromosome. Okay, and due to that the exchange, recombination, it will be occurs, which will be results into variations, and variations, it is the raw material for evolution. So for variation and evolution, meiosis, it is the important process. It will create variations and evolution. So that are the significance regarding with a meiosis. So finally, students, we have to study about significance of meiosis. First significance of meiosis is for formation of gametes. So due to meiosis, we know in sexual reproduction there is formation of gametes, male gamete and female gamete, which it will be undergo fertilization, form zygote, and in this way maintain diploid condition of the zygote. Okay, so meiosis and fertilization is the steps where there will be maintenance of constant chromosome numbers. Okay, so gamete formation के लिए, okay, क्रोमोजोम नंबर ऑफ स्पीसीज को मेंटेन रखने के लिए, कांस्टेंट रखने के लिए. थर्ड वन एप्लीकेशन वी कैन से स्टूडेंट्स, इफ देयर विल बी एब्सेंस ऑफ meiosis, देयर विल बी फॉर्मेशन ऑफ abnormal tetraploid or hexaploid cells. Okay, so such type of the cells, they will be abnormal. Next one application, student, in meiosis we know one of the very important event which will occur, it is a crossing over. So during crossing over we know there will be exchange of homologous segment of the chromosome. Okay, and due to that the exchange, recombination, it will be occurs, which will be results into variations, and variations, it is the raw material for evolution. So for variation and evolution, meiosis, it is the important process. It will create variations and evolution. So that are the significance regarding with a meiosis.